Thank you, Young. And I guess one of the benefits of organizing a session or an event like this is that I get to inflict new data and concept for feedback. So as Young mentioned, I'm going to be talking today about a new project in my lab that has really been driven by a new graduate student, Chad Jibo, who has a poster today. So if you want to learn more about the specific topic, please definitely go and ask him. So, disclosure. So, what I'm going to be talking about today is hopefully a slightly provocative look at or reinterpretation of immune-elicited or immune-mediated enhancement of dengue virus infection. I think we are all aware of the role that monocytes and other monocytic phagocytes are thought to play as the dominant cellular host or dominant cellular location of dengue viral replication. You can look at any textbook or any review or any introduction to a dengue paper, mine included, where you see that monocytes, especially monocytes, macrophages, and dendritic cells are the primary target of dengue infection and dengue replication. This is substantiated and supported by decades of incredibly robust work, including autopsy studies, analysis of cells isolated ex vivo from individuals or animals that have been infected with dengue, and also these are definitely the populations of cells that are most infectable ex vivo. In our hands, monocyte-derived macrophages are just about the only human cell that you can infect directly ex vivo. But it also fits a lot of the models of immune-mediated enhancement that we've talked about and we are continuing to talk about, in fact, in light of the fact that they express both the scavenger receptors that are thought to primarily facilitate dengue infection, dengue uptake, as well as expressing high levels of high-affinity Fc receptors that can facilitate antibody-dependent enhancement. However, if you go back in the literature and actually look at studies that have examined which population of cells in circulation during acute dengue harbor the most infectious virus, a slightly different picture emerges. Looking back in the 90s, the first instance I could see of this was a paper by Bruce Ennis, again coming out of AFRIMS in Thailand, where they used a strategy where they isolated T cells, NK cells, monocytes, and B cells from individuals experiencing an acute dengue infection, and then used an interthoracic inoculation of mosquitoes to test the presence of infectious virus. In all the individuals that they analyzed, it was the B cell fraction of the PBMC pool that actually harbored the most infectious virus in these patients experiencing acute dengue infection. Furthermore, in a study, again out of Thailand, senior author Alan Rothman here, was demonstrated again, if you isolated PBMC from individuals that were experiencing acute, primary, secondary, DHF, DF dengue infections, again, the B cells were the population of PBMCs that harbored, in this case, the most genomic material. This is looking by PCR for viral RNA content in the cells. And also, in our own work using slightly more modern technologies, different technologies, we were able to see by single-cell RNA sequencing that B cells, again, and this is a patient, again, from Thailand that was experiencing a secondary dengue 1 infection, that B cells, again, were the primary cellular reservoir of dengue virus. In this case, we're looking at dengue viral RNA, both positive and negative sense. And I think most convincingly, recent work from Shreet's lab in using a combination of single-cell RNA sequencing, flow cytometry, as well as co-culture techniques demonstrated that in individuals, again, experiencing primary, secondary infections, DHF, DF, a whole variety of dengue spectrum, dengue severity, that, again, the B cells were the population of cells in the PBMC compartment that had not only the most viral RNA, but when you isolated these cells out and co-cultured them with susceptible cells, these B cells were, in fact, harboring infectious virus. So what's going on here? This doesn't really meet or match the model that we've all gotten familiar with and we've all really described. What we know about B cells is that they don't express the canonical entry receptors that we normally think about for dengue. They don't express DC sign, they don't express mannose receptor, they're not really professional scavengers. They don't really have that type of uptake and processing machinery. While B cells do express an Fc gamma receptor, the Fc receptor that they express, Fc gamma receptor R2b, when engaged by an antibody, when engaged with an IgG antibody, actually induces anergy and can actually induce apoptosis in the B cell. And in studies, this is previously published work, not from our group, the serum and monoclonal antibodies that can induce ADE in cell lines that express traditional Fc gamma receptors like U937 cells, B cells aren't susceptible to traditional ADE. However, what B cells do possess is a specialized mechanism for binding and taking up antigen, namely the B cell receptor. And this is, again, we're all familiar with this, this is the same antibody that the cell produces, but instead of being secreted, this is a splice variant of the same antibody that is expressed on the surface of the B cell receptor, of the B cell. And if you dive into it, the structure and the signaling downstream of the B cell receptor is strikingly similar to what you see when an antibody bound to an antigen interacts with an Fc receptor. You have the ability both to restrain the antigen-antibody complex to the surface of the cell, and you're able to induce internalization and activation of signaling pathways such as PI3 kinase, AKT, so forth, and et cetera. So what we set out to try to ask about a year ago or so, and this is building on maybe 5 or 6 years of speculation by my group and colleagues, was could cells that express a dengue-specific BCR, could dengue-specific BCRs function as a preformed ADE receptor complex, meaning that you can then facilitate infections of B cells in a highly antigen-specific fashion? So we set off with I think also a recurring theme with this story is that this was a whole series of experiments that shouldn't have worked, but worked, and well, hopefully this keeps going forward. So the first experiment that we started off with to try to adjust this hypothesis was, okay, what happens if you just force a cell that is normally not susceptible to dengue infection? What if you force this to express a dengue-specific BCR? And the system that we used, we started off with 293 T cells, which are normally very poorly susceptible to dengue infection, and we transfected them with either the canonical dengue entry receptor DC sign, a negative control antibody, this is an influenza HA-specific antibody, or one of 3 different dengue-specific monoclonal antibodies. And again, these are constructs that have the transmembrane, this is a transmembrane IgG, so this has a small portion of the transmembrane domain at the end of the antibody. And then what we did was, after overnight incubation to allow these cells to express the receptor, we co-incubated these with a dengue 1 reporter virus. Given that we were putting an antigen-specific receptor on these cells, we wanted to make sure that any readout that we were looking at wasn't just simply antigen or virus binding to the cell. So the readout that we're looking for, you'll only get a signal from the cells, you'll only get a fluorescent protein signal if the cell is actually infected. This is a reporter virus that was initially developed by Ted Pearson and then Alan Rothman optimized for this purpose. And just to cut to the chase, we were able to show that, okay, we can transfect these cells with these transmembrane IgG constructs. The constructs that bind dengue do bind to the dengue antigens. But I think what you can appreciate here is that when we chuck this RVP on 293Ts alone, there's absolutely no infection. If we throw it on a cell that's been transfected to express DC sign, you get a decent amount of infection. The influenza-specific antibody does not facilitate any degree of infection, but then if you have a cell that is expressing a dengue-specific BCR, we get robust infection at rates equivalent to what we see for cells expressing the canonical transmembrane, the canonical receptor DC sign. However, we did notice that there was some heterogeneity in the, we'll just go back here for a second. You can see there is some heterogeneity. Not all receptors, not all BCRs seem to be created equal in terms of their ability to facilitate infection. And what was really interesting was that if you then use the monomeric soluble IgG version of these same antibodies, there is a high degree of correlation between the ADE activity of these monoclonal antibodies and the ability of these BCRs to facilitate infection of cells expressing them. So the ability of the cells to be infected that express these receptors. However, I just want to point out this is not a universal mechanism of viral entry or viral infection. This seems to be something, at least in our hands at this point, limited to flaviviruses or dengue virus specifically, because if we use an analogous system to look at a cell that's been engineered to express an HCMV-specific BCR, we've used a couple different antibodies. This is the MSL-109 and the 13H11. This does not facilitate infection of HCMV, even though we can get infection with expression of the PDGFR canonical receptor. So at least in this very reductionist system, we've been able to show that we can facilitate infection of cells that are not normally susceptible to dengue by expressing a dengue-specific BCR on its surface. Now, this introduces some very interesting implications. What this may mean is that in dengue infection not only fundamentally alters the humoral antibody landscape, the antibodies are in circulation, but may alter fundamentally the susceptibility of your B cell pool to infection, because you've now gone and increased the frequency of dengue-specific B cells after dengue infection, and therefore increased the number of cells that might be susceptible to infection via this BCR-mediated infection. So what we sought to do was test this directly. Does a primary dengue infection increase the frequency of cells that are susceptible to dengue infection, specifically B cells? And to get to this point, we utilized samples from the Dengue 3 human challenge model that we performed here in Syracuse a couple years ago. And again, this is Stephen will be talking about this in much greater detail later tomorrow. But again, this is a model where we selected dengue-negative volunteers and exposed them to an attenuated dengue 3 strain. And all the subjects in this study seroconverted and had a period of viremia that resolved within 28 days post-infection. So what we did was we took PBMC from prior to infection, which was when there was no pre-existing dengue, no dengue immunity at all, and looked at day 28 and day 90 post-infection. And we took those PBMC and again exposed them to this dengue 1 reporter virus to see what is the frequency of dengue infectable cells in this individual pre and post-dengue 3 infection. And what we saw up here was that there was a highly significant increase in the frequency of dengue infectable B cells in these previously dengue-naive individuals after their dengue 3 infection. This is quantified across all participants in the study. However, this was selectively to the B cell compartment. We saw no such change within the T cells and no such change within the monocytes in these individuals that experienced a dengue 3 infection. So there is a selective and specific increase in the infectability of B cells after you've experienced a dengue infection. So what we think we've demonstrated is that the expression of the dengue-specific BCR increases the susceptibility of normally non-susceptible cells to dengue infection. In flavivirus-naive individuals, there are very few dengue-susceptible B cells in circulation, but this number increases after dengue infection, after a primary dengue infection specifically. So one of the questions we wanted to know is okay, what is the phenotypic consequence of this dengue infection? What is actually happening to a B cell that's infected? We know a lot about what happens with monocytes and macrophages after you infect them with dengue, but what happens to a B cell when it's infected with dengue? And the problem is that the frequency of dengue-specific B cells in circulation is vanishingly small. So trying to tease that apart using dengue-specific memory B cells from PBMC pools was going to be very challenging. So what we wanted to try to do is find a way that we can reliably infect B cells in a BCR-dependent fashion without those cells having to express a B cell receptor, a dengue-specific B cell receptor, that is. So again, this experiment certainly gets the award for the most harebrained experiment that should not have worked. But what we did was we utilized a system where we cross-linked a dengue-specific antibody onto the surface of a B cell using an antibody specific for immunoglobulin. So in this case, the first experiment that we started off with was using the Raji B cell line, which I think a lot of us have used for various experiments. This cell is not normally susceptible to dengue, but is very permissive. If you transfect this cell line with DC sign, it is readily infectable, but without any additional manipulation, Rajis are not susceptible to dengue infection. And what happened is if we cross-linked dengue to a dengue-specific antibody and then cross-linked that antibody to the B cell receptor, in that specific situation, we were able to facilitate dengue infection. And again, it's only when you put all those cross-linking pieces together are you able to selectively facilitate infection of B cells. We were able to see this in not just B cell lines, but also in primary human B cells, where we were able to achieve pretty respectable infection rates in PBMCs from dengue-naive individuals with this cross-linking scheme. And interestingly, we were we're just starting to tease this apart, but the cytokine response to dengue infection in B cell does appear to be distinct from what we see in monocytes or macrophages, in that weirdly we get a lot of IL-8 production from dengue-infected B cells. If anyone has any thoughts about why that might be happening, I'd be very interested here. So again, I wanted to just emphasize, like I think as Ernesto said, what is old is new again, or what is new is old again, I don't know. But this result kind of harkens back to, again, an older paper from this case from Francis Innes, where he was able to demonstrate that simply cross-linking or restraining a dengue virus in an analogous cross-linking fashion to the surface of a cell was sufficient to facilitate infection. And I think this is a kind of a feature of dengue and flaviviruses in general, in that there's not a canonical receptor-ligand interaction like we have for SARS-CoV-2 or any of these many other viruses required for viral genome release. Simply getting a virus into an endosome, an acidified endosome, is enough to facilitate infection. So, which is why I think this BCR-dependent infection method or pathway is potentially intriguing and very potentially impactful. So just to summarize what I've talked about today and what we've worked on for the last little bit is that, again, expression of dengue-specific BCR increases susceptibility of cells to infection. In individuals that have no pre-existing dengue immunity have very few dengue-specific B cells in circulation and very few dengue-susceptible cells in circulation. Dengue-infected B cells may exhibit a different inflammatory profile than infected monocytes and macrophages. And we're proposing that this process of BCR-dependent infection, BDI, or BCR-dependent enhancement, BDE, whatever term we approve, represents a novel mechanism of immune-mediated enhancement that might fill a few gaps in our models of dengue immunopathogenesis. And I just want to point out where we think this might fit in. We've heard a lot of great talks today, work from Derek, Leah Katzelneck, Henrik Salge, showing that there's a very discrete window of antibody titers that are associated with enhanced risk of infection or enhanced risk of disease during secondary dengue infections. But again, not everyone who's infected with antibodies within that titer, within that range, develops severe dengue. So there has to be other factors, have to be other variables in play. And what we'd like to suggest is maybe the frequency of dengue-specific memory B cells or dengue-susceptible memory B cells is another variable in that equation, where if you have high antibody titers, that trumps all, you're not going to be infected. But as your antibody titers start to wane, the more stable population of memory B cells may actually represent a bit of a liability, because you have this population of cells that are now expressing a dengue-specific entry receptor that are just poised to become infected and produce virus quickly after exposure. So obviously this is a very nascent project, lots of unanswered questions, and I'd be excited to hear what other things we may have not thought about, but just some future thoughts and directions that we're keeping in mind is what is the relationship, obviously, between the frequency of dengue-specific B cells and the probability of developing severe disease. As I mentioned, a lot of these, we see a high frequency of dengue-infected B cells in circulation during acute dengue. What fraction of those are actually expressing a dengue-specific BCR? By this model, a lot of them should be, but that seems like maybe a bit of a high lift given the frequency of memory-specific cells that you normally see, dengue-specific memory cells in circulation. And finally, and potentially most intriguingly, what does this mean for the development of dengue-specific immunity, especially after sequential heterotypic infections? What I think we've realized as a community is that the immune profile elicited by two sequential heterotypic dengue infections may be different than what you can achieve with a simultaneous tetravalent administration or tetravalent vaccination event. Is that qualitative difference in two sequential infections because that second infection event is actually resulting in the infection and elimination of some B cell clones that were generated by your first infection? And again, I want to point out that the BCRs that were most capable of facilitating infection in this model were those cross-reactive fusion loop-specific antibodies. So perhaps a secondary infection is selectively infecting and pruning out memory B cells that express these undesirable cross-reactive, potentially enhancing antibodies. So with that, I want to thank everyone who actually did this work. I have a fabulous group, and I again want to shout out to Chad Jibo, who just passed his qualifying exam last week and is now back in the lab and hoping to tackle a lot more of these questions moving forward. And thank you.